Hello everyone, I'm Lode and today I'm going to talk about my tool, the AutoMLST. So what is the AutoMLST? AutoMLST is a tool that creates a multi-locus species tree. These trees are used for the understanding of evolutionary backgrounds of bacterial isolates, which have a lot of applications for research. Currently, there are a lot of genomic data that can be used to yield accurate species designation via modern phylogenetic methods at multiple genetic loci. However, these often require extensive expertise and time. That's why the automated multi-locus species tree, or AutoMLST, was created to provide a rapid one-click way to simplify this workflow. The second thing we're going to talk about is how does the AutoMLST tool work. The AutoMLST utilizes a multi-locus sequence analysis, or MLSA, to produce a high-resolution tree and thus does not produce a multi-locus sequence typing, or MLST, which is a related classification method. The phylogenetic tree also includes helpful annotations such as species class designation and secondary metabolite counts, which are handy for drug research. AutoMLST can automate selections of reference genomes and outgroups organisms based on one or more query genomes. Because of this, a wide range of researchers can perform rigorous phylogenetic analysis more rapidly compared to manual MLSA workflow. And now the question, why is AutoMLSA created? As said earlier, AutoMLST is a tool that uses MLSA to produce a high-resolution tree, but previous other methods were used and MLSA is the advanced version of them. Some of the earlier methods were horizontal gene transfer, or HGT, and that served as a powerful tool to discriminate species with the use of DNA-DNA hybridization methods, or DDH. This method is supplanted by genomic sequencing of conserved areas such as the 16S ribosomal DNA sequence presented in all bacteria. The problem with this method is that the complications such as using partial 16S sequences or multiple variants can be a source of misleading designations. Methods as the average nucleotide identity, or ANI, or in silico DDH have provided additional similarity methods using whole genome data. The problem with the similarity-based approaches is that it's difficult to interpret when no close relatives exist in the current database. A solution for this is the use of phylogenetic methods. Initial implementations include similarity-based tree construction. However, these do not take into account parameters of evolution, such as high rates of transition compared to transversions. Maximum likelihoods are alternatives that include these evolutionary parameters and often yield more accurate results over similarity-based approaches. Unfortunately, these variety of processing techniques discourage widespread use. At best, practices are not immediately apparent to non-specialists. This is reduced by web interfaces that utilize the computationally expensive maximum likelihoods approaches. These assure high confidence in evolutionary reconstruction, as a choice of model can give varied results, but this process can often be insufficient in categorizing species splits using the 16S data alone due to limited phylogenetic signal in the highly conserved sequences. A solution to this is the use of multi-locus sequence analysis, or MLSA, a technique that integrates many genomic loci to increase phylogenetic signal. By analyzing many conserved genes, often including the 16S data, a high-resolution species tree can be inferred. These advantages come with a cost of computationally expensive workflow with an obscure set of options and processing steps. Thus, automated multi-locus species tree, or AutoMLST, was created to simplify this progress and make it more user-friendly. And now we're going to have a look at the tool. So now that we are at the tool, we can have a quick overview of the tool. So first we see a rapid ANI estimation, or average nucleotide identity. Easily explained, this is a measure of nucleotide level genomic similarity between the coding region of two genomes. The second thing we see is the MLST gene selection. And we can read the single copy gene with dn divided by ds value lesser than 1 are identified and prioritized. And this number will tell us something. If a ratio is greater than 1, we have a positive or Darwinian selection. If it's lesser than 1, we have a purifying or stabilizing selection. And if the ratio is exactly 1, we have a neutral selection. And then we have tree building, but we will explain it when we get to it. So we click Start Analysis, and we have two options, the de novo mode and the placement mode. The placement mode is a fast reference tree is fixed, and the unknown query sequence are placed onto the tree via a reference alignment. Then we have some options that we can fill in, but these are the same with de novo mode, so we go directly to that. And de novo mode is a method for peptide sequencing performed without prior knowledge of the amino acid sequence. And then we have the options that you can overlook. So the first thing is you can paste your NCBI accession number here. So we'll get our accession number directly from NCBI and paste it here and upload it. You can do something else, and that is you can upload a file with your FASTA sequences. This file cannot contain more than 20 genomes, and the genomes need to be of the same organism. Then you can also upload it, but we will use an NCBI accession number. You can also paste your email address and a title of your job here. Because this will take a while, we can send it to our email so that we don't need to wait here the whole time. And then we have some options. First, you can select to skip step 2 and step 3. If you skip step 2, you cannot select manually your organisms, and it will just pick the 15 nearest organisms. But if we don't mark it, we can do it ourselves, and I will show it to you. It's the same with step 3. If we select it, it will automatically pick the 100 closest MLST genes, but if we don't select it, we can choose them ourselves. Also a thing, if you don't select this, you can also manually select your outgroups. So that's a thing that we also can do. Performing an IQ bootstrap. A bootstrap is the procedure when you take a random subset of data and rerun the phylogenetic analysis, and the reported value is the percentage of bootstrap replicates in which the node showed up. So if we have, we do this 1,000 times, and if a certain sequence or a certain part of the branch shows up 900 of the 1,000 times, it will have the number 90 with it, because 90% of the times these two branches are together. So we will select this, that we have this, and then the run model finder, this just finds the optimal model for your tree, so we can also select this. And then filter inconsistent MLST genes. This is just to have a smoother process, that we don't have some false MLST genes that are annoying to work with. And then the fast alignment mode of the MAFFT, FFT, NS2. This is a fast alignment mode, and the FFT NS2 is an alignment for sequences under 300,000 nucleotides. And then we have save link and cookies, but it's just for if you want to have this browser quick back, because if you don't let it go to your email, you want to get back here. And then we have the concatenated alignment and the coalescent trees. And phylogenetic trees, you have two fundamental different ways to build it. In one gene sequence are concatenated into a supergene alignment, which is then analyzed to generate the species tree, and the other phylogenies are inferred separately from each gene, and then consensus of these genes phylogenies is used to represent the species tree. So we will do the concatenated alignment, and we'll get our accession number from NCBI. So if you go to NCBI, I already looked it up, and we take a sequence of a bacteria, we can copy it and paste it here and upload it. It will take a few minutes, and I will go back when it's done. So now you can see our sequence is uploaded, and we can go through with it. We click submit job, and we wait till the first step pops up. So now you can see this first step is loaded, and we can select 50 organisms that we want. So there are 50 selected by the program itself, and we can add some extra that are here, but we will leave this the same. And then we have the outgroups. Now you see that there is one outgroup, but we can add some more. So if we add like 5 and add the list, we have now 5 outgroups, and we will continue and wait till the next part. So now we are at step 3, and we can select our MLST genes. Again, the program has selected the 100 best for us, but we can do it manually if we want. We can put some extra in or some genes out, but we will leave it how it is. And you have the overview, and you can choose again to change the tree output. We will leave it at concatenated and continue. So after some time, the tree has loaded, and you can see we have our tree. Now we are going to sum up what everything means. First, we see that some dots have colors. We see the blue dots has a QS annotation, and this is our sequence that we put in, so that we submit it, that we extract from the NCBI gene browser. Then we see the red dots, here 4 and here 1, and these are the out groups. This is also marked with OG. So this was the out group that was automatically selected, and these are the 4 that we have selected. So if we didn't select these, we would have another routing. So now our routing is here at this point, but our routing would be different if we only used this one as our out group. Then we see the numbers, the 1, the hundreds, and all. And these are the bootstrapping that I explained. And then we have the green dots. The green dots stand for type strain. And this is used when the species was described for the first time, and which was selected by the author as a type strain. Then we have some options that we can do. We can shove the tree to left or right with these 2 buttons with 100 pixels per step. And this is handy if we toggle on the scaled or the unscaled branches. Now it's unscaled. If we click on this, it's scaled, and you see that the tree is cropped. But if we press enough on the plus button, we will see a scaling, but it will take a lot of our screen. So if you want to do it yourself, you can, but I won't do it because of time. Then we have the display any groups. Then we can have a cutoff value, like if we have a 97% cutoff value, we have some groups, and we see where the cutoffs are. So you see this is one group, the green ones, and you have the purple ones, the pink ones, the blue ones, and so on. And you can do this with the different cutoff values. Then you have the strain info. This is what I explained, the query sequence, the outgroups, and the type strains. And then you can just reset the colors. So if we toggle this back on, and this is what you have, and here you have all the data and all the sequences that were selected. Here you have again the most similar organisms that the program gives to you. These are the 10 most, but you can go further and look for the other ones. And then you can download the tree if you want it. So this was the tool, and I hope you learned something from it. Thank you.